environmental toxicology, otherwise known as ecotoxicology. So yesterday we were able to define some terms in toxicology, talked about toxins, talked about toxicity. We tried to link toxins and toxicity together. And then we were able to establish the fact that toxicology deals with the study of poisons and how it affects the tissues and organs of animals. All right, so we looked at that. We looked at the various branches of toxicology. We looked at the clinical toxicology. We looked at agrotoxicology. We looked at industrial toxicology. We also looked at environmental. We looked at veterinary toxicology and the rest of them. And then we also looked at mode of toxic actions. We said these toxicants, they gain entrance into the body via injection, absorption, and inhalation. So once the organism is exposed to these poisons through these four methods of entrance, these chemicals get into the body. So ingestion, we talked about ingestion through the mouth, absorption and injection through the skin, and then inhalation through the nose. So when these poisons get into the body, they are being metabolized by the pancreatic amylase present in the small intestine, and then they get absorbed into the bloodstream through simple diffusion. And once it gets into the bloodstream, through active transport and facilitated transport, these poisons are being distributed all over the body. And then it begins to affect some of the organs and tissues in the body, begins to break them down. So we talked about that yesterday. So today we're going to quickly go to the history of toxicology. Much is not known about the history of toxicology because according to research, according to documentations, a lot actually was lost, especially in the early times. So there's no much documentations or manuscripts relating how toxicology came about. But we have it in the record that a German scientist by plotting the concentration against the dosage. All right, so let's look at the exposure classes of these toxins. So they include how are we exposed to these toxins. Number one, we have them in our food. Exposure classes include toxicants in food. Many of us have experienced food poisoning before. Yeah, that one is even a very low grade of toxicity. So you can find them in food when somebody mistakenly slips a poison into your food. Just maybe he has an agenda or something. So you can be exposed to toxins through the food that you eat. You can also be exposed to toxins through the air that you breathe, especially when the air contains those toxic compounds or gases. The more you inhale them, the more you're exposed. You can also be exposed to these toxins through the water that we drink. So once the water is polluted or contaminated, it becomes toxic. You can also be exposed via the soil. All right, the soil. How? Let's give example. When the soil is contaminated, one of my students is actually carrying out a research on that, the effect of diesel oil contamination on the growth of zimi, that's maize. So he planted maize in a particular soil, and then we poured diesel oil on that particular piece of soil, that particular land. Now everything percolated inside the soil. And remember, as the plant is growing, the xylem of the roots will usually go down the soil to pick up nutrients. and then transfer it to the xylem of the stem, and then xylem of the stem will not transfer it to the xylem of the leaf, from which photosynthesis takes place. All right. So when after photosynthesis is completed, the flame of the leaf transfer the already manufactured food, which is the starch, back to the flame of the stem and then to the flame of the root. So you see that the food is not distributed across the whole plant. Now imagine when the xylem of the roots trying to get the nutrients from the soil, which is usually so. In order to achieve these three objectives, which is obtaining data, meeting the legal requirements, and developing empirical or theoretical principles. In order to achieve these three objectives, the main areas of study include, number one, the distribution of pollutants in the environment. So how are these pollutants distributed in the environment? What is the entry mode? How do they move from one ecosystem to another ecosystem? Are they stored? Are they transformed within the environment? So all of this helps us to, these are some of the reasons why we collect data about these chemicals, just to understand their distribution in the environment, how they enter the environment, their movement, the storage, and how they are transformed from one compound to another compound, or how they are being splitted from compounds to simple elements causing problem. So that's one of the major things to be able to achieve them. The number two, at an individual level, toxicants may disrupt the biochemical, molecular, and physiological structure and function, which will in turn have consequences for the structure and functions of the communities in the ecosystem. So these toxicants at individual level, we need to understand the everything that happens when it gets into the system. That's what I mean by biochemical, molecular, and physiological structure and function. Because once it begins to affect the individuals in the community, it begins to affect their functionality. They won't be able to function effectively in the community. They won't be able to contribute to the food chain, okay, within the ecosystem. Many of them will die, and there'll be a reduction in the population. All of this describes the function that they play within the communities within the ecosystem. And then number three, at the population level, it may be possible to detect changes. That is the number of individuals. So when the population decreases from 100 to 50, something is actually going on. So it helps us to identify that. In gene frequency or changes in ecosystems which are attributable to pollution. So all of these things is what ecotoxicology achieves. All right. Then for us to understand ecotoxicology, for us to be able to study ecotoxicology, it is possible to use biomarkers. Okay, biomarkers to establish that the natural population has been exposed to pollution, and this can provide a valuable guide to whether or not a natural population is at risk or in need of further investigation. So we use biomarkers to detect how much these toxins have entered the population. So are they at risk, or it can be dealt with? So it gives us valuable information. Biomarkers give us valuable information about what is happening in a given population in relation to toxins. Are they at risk, or it's manageable or tolerable, or do they need to leave? Is there any need for people to begin to leave that particular given ecosystem? So biomarkers is also used to achieve that. For the purposes of regulation and registration of chemicals, the toxicity of individual chemicals is principally investigated via toxicity testing. So before you come up with a chemical they want to use for an industrial process or any kind of process, it has to go through what we call toxicity testing, so that we understand the dose response relationship and how it may even affect it. So the main tool used for toxicity testing is what we call the standard toxicity test, the STT, which usually tests the dose or concentration of a particular chemical that is toxic to under controlled laboratory conditions. So that's what ecotoxicology also tends to achieve. Now, these toxicity tests are mainly carried out using individual animals, although there has been a move towards the use of more complex systems known as the mesocosms. All right. So for us to understand toxicity tests, we usually use a particular animal to do the test. But like we're saying now, it is branching out to having to involve more animals at the same time, and systems that have complex nature. Like given ecosystem, you have distribution of different animals, not just a particular kind of animal, the big, the small, the very small, just different distributions of different kinds of animals in an ecosystem. Then we see how it affects individuals or each of those animals that make up the ecosystem. Now, in some situations, particularly in the case of pesticides, it may be possible to carry out field trial to assess toxicity. Yeah, field trial in the sense of you just distribute the pesticide, you just spray the pesticides in a given environment, and then see how much of the insects and pests and the rest of them are being affected by that. The data, toxic data, are used to make assessments of the hazard and the risk posed by a particular chemical. So after we've done that, we do a test. When we do the toxic test in a controlled laboratory setting, it helps us identify the level of risk in such chemical, and then it can now be either be approved for use, or there may be a regulation for its use if it has a high concentration. Significant issues with chemicals that have driven the development of ecotoxicology. DDT. We talked about the work that was done with DDT. This one has already spread around the world. Cadmium in Japan, mercury also in Japan, the PCB in Japan and Taiwan, the dioxins of various kinds. So all of these things have also helped to contribute in the study of ecotoxicology based on the effects they all have in the environment. All right, so importance of toxicity test. Let's quickly look at the importance of toxicity test. I'm trying to just see how much I can cover because today is your last class. So let's cover as much as possible. Importance of toxicity test. Number one, toxicity tests are used to expose test organisms to medium, water, sediment, or soil and evaluate the effects of contamination on their survival, on their growth, on their reproduction, behavior, and other attributes of the organisms. So toxicity test helps us to know the effect when organisms are contaminated with these chemicals. How does it affect their survival? How does it affect their growth? How does it affect their reproduction? How does it affect their behavior and other attributes? So that's number one importance of toxicity test. Toxicity test also helps to determine if the contaminant concentration in a media are high enough to cause adverse effects in the organization. If you give a low dose, it may have a low effect. For a situation where it's a high dose, it gives us a high effect. So toxicity test tells us at what particular dose, at what particular level does it become, does it have an adverse effect on the organism. Right, then toxicity test can also be used to check if cleanup of a site is effective. One of the ways we clean up all these chemicals in the environment is called bioremediation. So through toxicity tests, we know that if the bioremediation method or process that we employed is effective enough to clean up the chemicals in that particular environment. Because after you've done your bioremediation, let's say for example you're doing what we call mycological bioremediation, where you use fungi to actually kill some of these things. At the end of the day, you do a toxicity test to see whether the level is still high or is low. Then bioaccumulation test helps to know the substance that can bioaccumulate in the tissues and organs of organisms. So there are some that once they enter into the body, it just goes to the various tissues and organs and then accumulate there. Those ones may not give an instant effect, but over time it begins to have effect. So those are some of the importance. There are other many importance of toxicity test. Then types of toxicity test, there are two major classes of toxicity test. We have the acute and the chronic, right? It can be measured. Toxicity test can measure acute and chronic effect. When we say acute, it is not, it is really, it's not too, it's not, it's happening for a short period of time, but when it's chronic, it becomes for a very long period of time. All right. So acute toxicity is a discernible adverse effect induced in the test organisms within a short period of time, a short period of exposure. While the chronic is for a long period of exposure. That's for the chronic. Okay, I think we'll stop here for today because my time is really up. There are other things we would have looked at because of time. Toxic testing and the regulation and release of toxic chemicals. I think I've said a little about it, so we already have an idea of toxic testing and then what kind of regulations should be given before these toxic chemicals are allowed to be used within the environment for whatever purpose that is used for. Okay, so I think we'll stop here. I'm trying to see if there's anything I have in my slide that may be important. Okay, I think what I just have left is just a sample of how to do a toxic test where I had to use fish, yes, to just carry out. So you have your parameters, the methodology that I use just to explain how toxic test is done. We have an apparatus, how data was collected, the animals that were used, fish, shrimp, earthworm, how they were acclimatized because we had to remove them from the water and then place them in a new environment, how they were able to acclimatize to the new environment, then how the chemicals were actually injected into the aquarium at different levels. We had from 10 to 100 mil, and then it was actually observed for a period of 8 to 24 days. Then at the end of the day, we're able to harvest these fishes and then begin to harvest some of the tissues and organs. And then we now took them to the lab to do a bioassay to identify the kind of defects that we had. Then we're also able to record mortality rates of some fishes because some fishes could not survive it. They died even before the 28-day mark. Some of them died. We're able to take a record of the time of death in cognizance with the amount of the level or dosage of that particular chemical that was given to that particular fish that died. So that will now tell us the tolerance level of that particular fish to that level of toxin. Right. So I think that's that. Then the rest are statistical analysis just to explain the results, then the interpretations and the rest of them. All right. Thank you so much for joining me in this class. I hope you enjoyed yourself. And then until I see you again, do have a wonderful day. Bye for now.